History of DNA. What is DNA and how was it discovered? The human hereditary material known as deoxyribonucleic acid, or DNA, is a long molecule containing the information organisms need to both develop and reproduce. DNA is found in every cell in the body, and is passed down from parent to child. Although the discovery of DNA occurred in 1869 by Swiss-born biochemist Fredrich Miescher, it took more than 80 years for its importance to be fully realized. And even today, more than 150 years after it was first discovered, exciting research and technology continue to offer more insight and a better answer to the question, why is DNA important? What is DNA made of? How does DNA work? Who discovered DNA? When was DNA discovered? DNA is self-replicating material that's in every living organism. In simplest terms, it is a carrier of all genetic information. It contains the instructions needed for organisms to develop, grow, survive, and reproduce. It's one long molecule that contains our genetic code, or recipe. This recipe is the starting point for our development, but DNA's interaction with outside influences such as our lifestyle, environment, and nutrition ultimately form the human being. While most DNA is found in the nucleus of a cell, a small amount can also be found in the mitochondria, which generates energy so cells can function properly. Perhaps the most fascinating part of the process is the fact that nearly every cell in your body has the same DNA. What is DNA made of? DNA is made up of molecules known as nucleotides. Each nucleotide contains a sugar and phosphate group as well as nitrogen bases. DNA's structure is a double-stranded helix, and it resembles the look of a twisted ladder. The sugar and phosphates are nucleotide strands that form the long sides. The nitrogen bases are the rungs. Every rung is actually two types of nitrogen bases that pair together to form a complete rung and hold the long strands of nucleotides together. Remember, there are 4 types of nitrogen bases, and they pair together specifically: adenine pairs with thymine, and guanine with cytosine. Human DNA is unique in that it is made up of nearly 3 billion base pairs, and about 99% of them are the same in every human. However, it's the sequence of these bases that determines what information is available to both build and maintain any organism. Think of DNA like individual letters of the alphabet. Letters combine with one another in a specific order and form to make up words, sentences, and stories. The same idea is true for DNA. How the nitrogen bases are ordered in DNA sequences forms the genes, which tell your cells how to make proteins. Ribonucleic acid, RNA, another type of nucleic acid, is formed during the process of transcription, when DNA is replicated. RNA's function is to translate genetic information from DNA to proteins as it is read by a ribosome. How does DNA work? DNA is essentially a recipe for any living organism. It contains vital information that's passed down from one generation to the next. DNA molecules within the nucleus of a cell wind tightly to form chromosomes, which help keep DNA secure and in place and store important information in the form of genes to determine an organism's genetic information. DNA works by copying itself into that single-stranded molecule called RNA. If DNA is the blueprint, you can think of RNA as the translator of instructions written in the blueprint. During this process, DNA unwinds itself so it can be replicated. RNA is similar to DNA, but it does contain some significant molecular differences that set it apart. RNA acts as a messenger, carrying vital genetic information in a cell from DNA through ribosomes to create proteins, which then form all living things. How was DNA discovered? DNA was discovered in 1869 by Swiss researcher Friedrich Miescher, who was originally trying to study the composition of lymphoid cells, white blood cells. Instead, he isolated a new molecule he called nuclein, DNA with associated proteins, from a cell nucleus. While Miescher was the first to define DNA as a distinct molecule, several other researchers and scientists have contributed to our relative understanding of DNA as we know it today. And it wasn't until the early 1940s that DNA's role in genetic inheritance was even begun to be researched and understood. Who discovered DNA? The full answer to the question who discovered DNA is complex, because in truth, many people have contributed to what we know about it. DNA was first discovered by Friedrich Miescher, but researchers and scientists continue to expound on his work to this day. as we are still learning more about its mysteries.
As it turned out, Miescher's discovery was just the beginning.
Credit for who first identified DNA is often mistakenly given to James Watson and Francis Crick, who actually just furthered Miescher's discovery with their own groundbreaking research nearly 100 years later. Watson and Crick contributed largely to our understanding of DNA in terms of genetic inheritance, but much like Miescher, long before their work, others also made great advancements in and contributions to the field. 1866, before the many significant discoveries and findings, Gregor Mendel, who is known as the father of genetics, was actually the first to suggest that characteristics are passed down from generation to generation.
Mendel coined the terms we all know today as recessive and dominant.
1869, Friedrich Miescher identified the nuclein by isolating a molecule from a cell nucleus that would later become known as DNA. 1881, Nobel Prize winner and German biochemist Albrecht Kossel, who is credited with naming DNA, identified nuclein as a nucleic acid. He also isolated those 5 nitrogen bases that are now considered to be the basic building blocks of DNA and RNA: adenine, A, cytosine, C, guanine, G, and thymine, T, which is replaced by uracil, U, in RNA. 1882, shortly after Kossel's findings, Walther Fleming devoted research and time to cytology, which is the study of chromosomes. He discovered mitosis in 1882 when he was the first biologist to execute a wholly systematic study of the division of chromosomes.
His observations that chromosomes double is significant to the later discovered theory of inheritance.
Early 1900s, Theodore Boveri and Walter Sutton were independently working on what's now known as the Boveri-Sutton chromosome theory, or the chromosomal theory of inheritance. Their findings are fundamental in our understanding of how chromosomes carry genetic material and pass it down from one generation to the next. 1902, Mendel's theories were finally associated with a human disease by Sir Archibald Edward Garrod, who published the first findings from a study on recessive inheritance in human beings in 1902. Garrod opened the door for our understanding of genetic disorders resulting from errors in chemical pathways in the body. 1944, Oswald Avery first outlined DNA as the transforming principle, which essentially means that it's DNA, not proteins, that transform cell properties. 1944 to 1950, Erwin Chargaff discovered that DNA is responsible for heredity and that it varies between species. His discoveries, known as Chargaff's rules, proved that guanine and cytosine units, as well as adenine and thymine units, were the same in double-stranded DNA, and he also discovered that DNA varies among species. Late 1940s, Barbara McClintock discovered the mobility of genes, ultimately challenging virtually everything that was once thought to be. Her discovery of the jumping gene, or the idea that genes can move on a chromosome, earned her the Nobel Prize in Physiology. 1951, Rosalind Franklin's work in X-ray crystallography began when she started taking X-ray diffraction photographs of DNA. Her images showed the helical form, which was confirmed by Watson and Crick nearly 2 years later.
Her findings were only acknowledged posthumously.
1953, Watson and Crick published on DNA's double helix structure that twists to form the ladder-like structure we think of when we picture DNA.
When was DNA discovered?
What we know about DNA today can be largely credited to James Watson and Francis Crick, who discovered the structure of DNA in 1953. Despite there being many important and contributing discoveries both before and after their work, this is the year they discovered DNA's double helix, or spiraling, intertwined structure, which is fundamental to our current understanding of DNA as a whole. The future of DNA.
The future of DNA has great potential.
As researchers and scientists continue to advance what we know about the complexities of DNA and the insights it codes for, we can imagine a world with less and better managed disease, longer lifespans, and a personalized view of medicine that's specifically applicable to individuals rather than the population as a whole. DNA insights are already enabling the diagnosis and treatment of genetic diseases. Science is also hopeful that medicine will advance to be able to leverage the power of our own cells to fight disease. For example, gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a therapeutically beneficial protein. Researchers also continue to use DNA sequencing technology to learn more about everything from combating infectious disease outbreaks to improving nutritional security. Ultimately, DNA research will accelerate breaking the mold of the one-size-fits-all approach to medicine. Every new discovery in our understanding of DNA lends to further advancement in the idea of precision medicine, a relatively new way doctors are approaching healthcare through the use of genetic and molecular information to guide their approach to medicine. With precision or personalized medicine, interventions take into consideration the unique biology of the patient and are tailored individually to each patient, rather than being based on the predicted response for all patients. Using genetics and a holistic view of individual genetics, lifestyle, and environment on a case-by-case basis, doctors are better able to not only predict accurate prevention strategies, but also suggest more effective treatment options. We've come leaps and bounds from where we were in terms of our understanding of DNA 150 years ago.
But still, there is much to learn.
And with the potential that a deeper understanding of DNA will improve human health and quality of life across our world, no doubt, the research will continue. A full understanding of DNA of and between all living things could one day contribute to solving problems like world hunger, disease prevention, and fighting climate change.
The potential truly is unlimited, and to say the least, extremely exciting.